Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Hudash. Hello, everyone, and welcome back to another episode of our Jack Westin MCAT Podcast with your hosts, Mike and Molly. Mike, I'm really excited for this episode because I feel like we've been building to this. And in fact, maybe we even hit a crescendo last episode.
Yeah.
But this week we're putting it all together, which is really fun.
It's so cool because again, we've been kind of really diving into the main pathway of MCAT metabolism, which is aerobic respiration, starting with glycolysis, then weaving into PDC, TCA, kind of crescendoing up to the electron transport chain and oxidative phosphorylation. And while this is kind of the main pathway that we really focused on, there are so many other pathways that the MCAT loves to test on. But the reason I think it's so cool that we've really kind of gone in depth these last few episodes is because all the other pathways really connect to the central pathway, which I think is so cool. And really, when we teach this, I know when we both teach this, we always kind of teach it not to just think of these pathways as just like, oh, this is one pathway and now it's in its box, now it's on its own, or this pathway is in its box and on its own, that a lot of people actually learn in that way. But rather,
I tried it.
Yeah, I tried it. It doesn't work.
Everyone tries it.
Doesn't work at all. But rather, to be thinking about all of these pathways as just kind of one interconnected system. And I think that's our kind of goal today, is to kind of really focus on how certain molecules have different fates. It doesn't always have to go to that next step in, from glycolysis to PDC, depending on maybe the environment that the cell is in. Maybe it can do other things, and there's certain signals and certain, environments in the body that will choose some pathways over the other. It's not random. There's a lot of intention behind it, but because the body is very intentional, but the cell is, we have to learn it intentionally. We have to learn about the big picture so that we can understand when they ask us questions on the MCAT, like, oh, what will happen in this environment or in this case? We need to understand the connections between the pathway in order to answer these questions.
Yes, absolutely. And I feel like it's so beautiful to think about the body and how many different types of jobs it can fill, how many different types of things your body can do. And I mean, you can, maybe you know how to break dance and you know how to give a speech, right? But you're not going to do both of those at the same time. And you're not going to do, you're not just going to suddenly start doing either one of those. You're going to wait until you have the proper cues. You are responding to, in that situation, the environment around you and making sure you're picking something that's appropriate for the conditions that you're in, right? You're not going to get up on, a debate stage and do a breakdance, right? You're not also going to.
I mean, you won't, but
but maybe
I don't know.
we should.
I might, if I'm feeling kind of funny that day. But yeah, I get what you're saying.
You know what? Maybe you just win immediately. Who knows? But again, like your body, in terms of its metabolism, keep in mind we have catabolic processes that break things down to harness those raw materials or energy, and we also have processes that build things up. We have a lot of opportunities to break different types of macromolecules down, and we also have different opportunities to build different type of macromolecules up, right? We have so many possibilities. How can we decide what each cell should be doing at any given time? We need to get on the same page. We need to use the context of what's going on in the body to figure out which processes, especially catabolism versus anabolism, should be prioritized at any given point in time. So we're going to talk a lot about the control of these pathways, not like individual regulation of individual enzymes, but how the body can determine what's going on in its environment and how that's going to shape the different pathways that are going to become active. So I'm excited. We're going to be talking about a lot of the intermediates that we've discussed over the past 3 episodes on metabolism. And we're going to talk about how those molecules can ultimately serve as connection points between lots of different metabolic pathways. And if this idea sounds kind of foreign to you, know that we've already done that, right? We've actually already presented 2 such connecting molecules that were just connecting different phases of aerobic metabolism, right? Think about NADH and FADH2. Those were both molecules that are produced all throughout aerobic metabolism, right? They are not directly used in those processes. They're produced, but those molecules, NADH and FADH2, are really shuttled to the electron transport chain, and that's where they're valuable, right? Those molecules serve as a connection between these different pathways that ultimately helps you carry out your overall goal. So that's kind of what we're going to be doing, except perhaps on a slightly broader scale with lots of different levels of metabolism involved. I'm excited. It's going to be fun.
Yeah, no. So let's kind of set the stage then with, like, you mentioned something wonderfully before. We're talking about how there's a reason that the body uses certain pathways, decides which pathway to use at what time. And I was kind of like talking about how the body communicates with cells, how it gets all the cells in the body kind of on the same page. And that's using hormones. Again, that topic that we discussed. Oh my goodness, how many episodes was that ago? When we
Too
talked
many,
about
too
too many
many.
episodes. We talk about the endocrine system. If you haven't seen the endocrine system lecture, go check that out, where we kind of really dive into all those kind of endocrine hormones and pathways. But there's kind of two hormones, especially when it comes to metabolism, that we really want to be focused on. There are others involved, of course, but kind of two mainly that we discuss, which are insulin and glucagon. Right? Those really signal the kind of two states the cell or really the body is in: the fed state versus the fasted state. Now, I want you to think of fed state as like what you would think, right? Fed means I'm full, I'm happy, I'm digesting, right? And that is kind of more signaled by insulin, right?
Yes.
It's when you eat, right? When you're eating, you got a lot of sugar, your body wants to uptake that sugar from the blood. It's kind of a review. The goal of insulin is to reduce blood sugar, to take up those molecules into the cells. But if I'm taking a bunch of sugar, well, probably if I'm well fed, I probably don't need to be busting out a lot of energy at that point. So I'm probably going to be wanting to store that energy, is more the fed state. And there are certain pathways that are very much favored in the fed state versus not favored in the fed state. But then if we have a fed state, we also have conditions of a fasted state. Think of a fasted state where, imagine you haven't eaten in a day, maybe a few hours, you're getting kind of hungry, like, when your belly starts rumbling. Well, that's when we don't have a super excess of energy. Well, that means we need to provide energy for the cell. We maybe even need to up the blood glucose because we don't have enough blood glucose there. And that's where glucagon comes in, right? And I think, Molly, you were the first one that mentioned this, and it was like, oh, that's such a cool way to think about it, that glucagon is released when glucose is gone. When there's no
I can't believe
glucose.
you haven't heard that
I
before.
had never
it's in
heard
the
of that. I
name.
just always kind of was like, oh, glucagon, cool. But like, yeah, when glucose is gone, you're in a fasted state, you release glucagon in order to re-bring that glucose back to kind of, you're in a fasted state. Let's bring that glucose. Let's mobilize some energy for the body to use. But again, in this fasted state, there are some pathways that are much more upregulated and some pathways that are much more downregulated. And when we talk about like these kind of pivot molecules, the molecules that make connections, a lot of the times the pathway that they follow is really dependent on the external stimulus, or rather, are we in a fed state or in a fasted state? And that's how we want to kind of be really prefacing today, to not memorize things. Don't try to memorize a specific pathway like, oh, this does this. Think big picture. Think, does this make sense? Given this environment, would the body want to store energy? Would it want to mobilize energy? But that's kind of prefacing kind of those external signals that the body's going to be telling the cell to know which pathway to follow.
Yeah. And I love talking about this because it calls back to our discussions of all the individual pathways, right? When we talk about insulin and glucagon, this is a organism-wide communication, right? These hormones traveling throughout the bloodstream, this is communicating to the body as a whole about the conditions that are present, right? And it's communicating with all the different cells of the body to change their activity levels, which I think is really cool because, again, you've got so many cells that make you up. They're all individually wrapped little bundles of membrane, right? Like they're all separate, but they all can communicate. And when we talk about things like insulin and glucagon, we should be thinking about things like phosphofructokinase, right? How it is regulated by insulin, right? And how ultimately insulin activating PFK-2 upregulates PFK-1, upregulates glycolysis. I like to just think, if I'm fed, if I'm full, I just had a huge meal, I've got a ton of energy. If there's been cell repair that I've been putting off because I've been hungry and haven't had energy, I'm going to go ahead and do it. If there's something that I've been meaning to do, I'm going to do it. I'm going to use the energy to do that. Upregulate glycolysis. Go ahead and use that ATP you produce through aerobic metabolism to do whatever you need. If I have excess, I'm going to store it because I've got a ton, right? When we think of the pathways that are upregulated, those are the ones that are going to come to mind when we're fed. And when we're fasting, right, or when we're hungry, it's it's, we're in lockdown mode. You don't want to
Right.
use energy unless you need to use energy. And we want to make sure we've got a ton of glucose for the cells that really need it, or the organs that really need it. So all of the pathways that we're going to discuss are going to fit pretty nice and neat into one of those states,
Kind of those
right?
boxes of like those two. What are we
Yeah.
doing, fed or
Are we fed
fasted?
or are we
Or
fasting?
right? And if you heard the things like PFK2 and
Yes.
phosphorodiockinase, and you're like, what is Molly?
Glycolysis,
What you're
episode
like, what
1.
is Molly talking about? Go watch our glycolysis episode where I think we like super broke down glycolysis like into like the
Yeah.
steps and like into an excruciating amount of detail because that's what the MCAT will expect us to understand. I think it's a good thing. I think it's a good thing.
I think so too. I think so
But
too.
so if you heard what Molly just mentioned, you're like, what's going on? I would pause the episode, go back, watch our metabolism. We just walked through all the metabolism and come back here, and then you'll be ready to go.
Yes.
Mhm.
Okay, I feel like we're ready to kind of start with one of our first biomolecules.
Yeah, like
Right?
the
Our
first like
first,
pivot point kind of deal, right?
yeah, and it
Mhm.
it actually comes from glycolysis.
Right, which is kind of the first step of aerobic respiration. Makes sense that that's where the first pivot point is,
Yeah.
right?
So again, we've got glucose. Let's say
Mhm.
glucose enters the cell. We talked about way back in part 1 of metabolism, we talked
Mhm.
about how hexokinase is going to add a phosphate group and we're going to make glucose 6 phosphate. This is the
Mhm.
first intermediate or
Mhm.
product of glycolysis.
Mhm.
Glucose 6 phosphate.
Mhm.
So glucose with a phosphate on it, 6 carbon.
Mhm.
This is actually obviously produced very early, but it's actually a very important
Extremely.
initial hub of activity
Mhm.
where we know, again, hexokinase, part of its job, that first enzyme of glycolysis, is to trap glucose in the
Mhm.
cell by
Mhm.
forming glucose 6 phosphate. From there, though, this molecule can take a bunch of different pathways.
Right, right.
Obviously, it can proceed through glycolysis. We know
Sure.
that. We love that. We talked about
Mhm.
that in episode 1 of
Mhm.
metabolism.
Mhm.
But it can also go a couple different ways, right? We
Mhm.
know that glycolysis is one of them. It can also be used to synthesize glycogen. That's
Mm.
called glycogenesis. And I
Mhm.
know glycolysis, glycogenesis, and glycogenolysis, they all sound very
So
similar.
very similar. Mhm.
Glycogen is our storage form of glucose, right?
Mhm.
Glycogenesis is generating glycogen. Okay,
Mhm.
so we could use that glucose 6 phosphate for glycolysis. We could use it for glycogenesis. We could
Mhm.
also use it for a pathway called the pentose phosphate pathway. And this is
Mm.
more to help us make, it's more an anabolic pathway. Okay, we
Mhm.
make things like nucleotides and we make a intermediate called NADPH, which
Mhm.
does redox reactions just like NADH,
Mhm.
although totally different context that this would be used.
Sure, sure. And I think it's really cool because we just kind of illustrate, first of all, very early on in glycolysis, the first kind of pivot point, because we know glucose 6
Mhm.
phosphate can now go through these three different pathways. And in order to
Yeah,
understand which pathway it's going to choose, let's think about the environment in the cell, right?
yeah,
We want to think about like, okay, if we're uptaking glucose from the blood, we're kind of putting a phosphate on it to trap it there, we're probably in more of a fed state, right? Insulin is really upregulated because again, we're taking glucose from the blood. But that's kind of the external thing that's happening. But the internal signals of the cell will also dictate what happens next. Because imagine we're in a fed state, but the cell itself doesn't have much energy. Like we're in like a fed state, and that cell's like, oh, I need energy because I don't
yeah.
have much. Well, of course, then it's going to proceed through glycolysis. It's going to help produce energy, like we just talked about before, produce the ATP. But what if it's happened for a little bit and now I've produced a whole bunch of ATP? I'm still in a fed state, but now I've got a whole
Mm.
bunch of ATP in the cell. Like, will I still want to be going through glycolysis?
No, right? Why would
Yeah.
you?
Well, I've got a whole bunch of ATP. Why would I do that? And it's one of the actually that key PFK step, like that PFK
Mhm.
enzyme, is actually inhibited by ATP, right? It actually says, okay, we might be in a fed state, but the cell, we don't need anymore. We're done producing ATP.
That's
Stop
the
doing
rate
glycolysis.
limiting enzyme of glycolysis, by the way,
Right.
PFK. We've mentioned it a
PFK.
couple times, just in case.
PFK-1. Call back to episode one
Go
of
back,
metabolism,
yes.
right? That rate limiting step, because this is the kind of the committed step of glycolysis. That's why this is the really regulated step of
Yeah.
glycolysis. So a bunch of ATP in the cell is going to be like, hey, all right, stop doing glycolysis. But then it's not like we're just going to have G6P sit around like it's not going through glycolysis. Well, we have other options because maybe the cell has enough energy, but why don't we just store some for a rainy day? Because we don't know we'll always be in this high energy state. Well, that's why G6P can actually be shuttled over to glycogen storage, where I say the word shuttle, but really it happens in the cytoplasm as well, where we just turn that glucose 6-phosphate into, via phosphoglucomutase, into glucose 1-phosphate and kind of get it ready and assemble it into glycogen, right? Where all it does is just store it for a rainy day, right? Because again, our cell has enough ATP, but it might not always be the case in the future. So now we've stored the glycogen, but here's the interesting thing is one, the cell can't store that much glycogen. Like there's only a limited amount of space in the cell, so it can store some, but then it'll stop storing eventually, right? It's not an infinite storage. But then so now we have enough ATP, we have enough storage. Okay, what else are we going to do with G6P? Well, that's where the third process comes in, which is the pentose phosphate pathway, which the pentose phosphate pathway, I think you really put it well, which is kind of it's good for like the stuff that we've been holding off. Like let's build some things. So we've got enough energy, our stores are settled. Let's start building things. So the pentose phosphate pathway is a really critical step to produce ribose 5-phosphate, ribose 5-phosphate, which is a very key kind of substrate for nucleotide synthesis. Yes, the same nucleotides for DNA and RNA. We actually use glucose 6-phosphate or this pentose phosphate pathway to produce those things. Because imagine, right? If I'm all good, I'm fed, let's start building things and things like DNA and RNA. So this is kind of a really cool thing to conceptualize. Like, yeah, based on where the cell is at, it might follow a different pathway from this glucose 6-phosphate. A cool thing from this pentose phosphate pathway is NADPH is also produced in this pathway. And again, I did not misspeak. It's NADPH,
Yes.
not NADH. Now, this is a very key distinction to make. Not the same molecule, not the same function, right? We went in on NADH, talking about how it's really important for energy production, the electron transport chain. NADPH, not for energy whatsoever. We do not use it for energy production in the electron transport chain. It's produced via reduction, like via redox reaction like NADH is, but its function is far different. It's much more used for cholesterol synthesis, for fatty acid synthesis, for protection against oxidative damage. Important functions, but very different functions. And so that's
anabolic for the most part.
Very anabolic.
Kind of catabolic, right? Yeah.
Right? And that makes sense because the cell has enough ATP, it's stored enough. And again, this is just a very anabolic process to produce the substrates for DNA and RNA production, the kind of get those molecules NADPH for fatty acid, for protein synthesis. Again, very building process, which makes sense considering the environment, the signals that are happening in the cell.
Yeah. I always think about it. I mean, NADH and NADPH, they're only different by a phosphate. That's why the P is in the name. But I always like to imagine, just in case you ever get it mixed up, that the P is for producing, right?
Cool.
NADH, but used for production. Again, they are both storing electrons. Like they are both reduced electron carriers. The difference is what they're going to use those electrons to do, right? Are we going to use it in a catabolic reaction where we're going to contribute to making ATP from basically glucose metabolism or metabolism of something? We'll talk about other options. Or am I going to use it to make something new? So NADPH is also the producing one, which is kind of nice. But yeah, cool. Again, glucose 6-phosphate is really it's our first product of aerobic metabolism, right? Again, we've got glucose, hexokinase makes glucose 6-phosphate, and we've already connected 3 processes. And of course, when we talk about glycogenesis, which is making glycogen, that would be what we do when we're well fed. You might as well go ahead and store glucose and glycogen. If we get hungry, we're going to go through the reverse, which is glycogenolysis. We're going to take glycogen and we're going to chop off, like you said, a glucose 1-phosphate, mix it around glucose 6-phosphate. We can now use that for glycolysis. And of course, whenever we talk about glycolytic intermediates, keep in mind you can do gluconeogenesis as well, which is the reverse of glycolysis. So really, we're sitting at the intersection of 5 processes, right?
So many, right?
Glycolysis, gluconeogenesis, glycogenesis, glycogenolysis, and the pentose phosphate pathway.
Oh my goodness, right? G6P is just so important, right? Where, I mean, the glycogenolysis is just the reverse of the glycogenesis, but yeah, that gluconeogenesis where I always kind of like thinking about it as like a reverse glycolysis, except with a few different steps,
Few
like
different enzymes,
a few
yeah.
different enzymes, specifically on the irreversible ones. But yeah, well, since G6P is one of the intermediates of glycolysis, well, it makes sense that it's also one of the intermediates of gluconeogenesis, which
Exactly.
I think is a great kind of connection between metabolic
Yeah.
pathways, which is what we're doing today. Which is what we're doing
Yeah.
today.
And again, I think it makes sense if you're fed versus you're fasting, what you would want to do in that case.
Right.
You want to prioritize the things that conserve energy or conserve glucose if you're hungry or if you're fasting. If you've got enough, go ahead and use it.
Mhm.
Okay, so that's G6P. That's our first one.
Mhm.
That being said, there are more, right? And
Mhm.
I would say the next major connection, other
Mhm.
than NADH, which we've talked about,
Sure.
the next one in our sort of aerobic respiration saga is
Mhm.
going to be pyruvate,
Mhm.
right?
Mhm.
Pyruvate is going to be another nice connecting molecule. This, of course, is the product of glycolysis. We
Mhm.
take one 6-carbon glucose and we make two 3-carbon pyruvates.
Mhm.
And pyruvate can be used in several different ways,
Very
right?
true.
We talk about how it could be oxidized into acetyl-CoA and then enter the TCA cycle. That's
Mhm.
assuming we have oxygen. Keep in
Mhm.
mind, if we don't have oxygen,
Mhm.
we need to continue with glycolysis alone. We're going to have to go into fermentation,
Mhm.
which again, we take NADH, we take the pyruvate that was just produced in glycolysis, we take that electron and that hydrogen, and we just shove it onto
Put on
pyruvate.
pyruvate.
We form
Shove it
lactate.
on is a great way to put
It
it.
basically does. I mean,
Just
close
push it
enough.
on. Yeah, yeah. And yeah. Mhm.
Yeah.
And I think that's really important and to kind of recall, like, yeah, like we need that when we're in anaerobic conditions because if we just kind of we can't go on and do PDC and all those steps. If we
Mhm.
just keep on doing glycolysis and anaerobic, we'll eventually run out of NAD plus. So that's kind of
Yeah.
the real key reason for fermentation is to kind of resupply or regenerate the amount of NAD plus in the cell. So by taking those electrons away back, take them back
Mhm.
from NADH, slap them onto pyruvate to produce lactate, and now the
Yeah,
NAD
I like that.
plus slide, yeah, just throw it on there, and then that NAD plus can go back into glycolysis to keep the system running in non-oxidative conditions. So that's
Yep.
already two connections right there, which we've already covered, which is really great. Like we already
I
talked about
know.
kind of those pathways. Like it can kind of
Mhm.
like pyruvate can go to aerobic respiration, it can do anaerobic, but those aren't the only things you can do,
Mhm.
is it? Right? What else can
Yeah.
pyruvate kind of flex into?
Well, the fact that the pyruvate molecules can make acetyl-CoA, that actually, acetyl-CoA, spoiler alert, we're going to go there next. There's a lot of
Oh
things
wow.
acetyl-CoA can be used to do.
Hmm.
Because pyruvate can be made into acetyl-CoA, we can kind of think it's got a lot of options too. But let's focus on pyruvate, the actual molecule, right? So
Hmm.
it can go to pyruvate dehydrogenase complex, make acetyl-CoA, and then do TCA cycle. For example, we could talk about lactate if we're
Hmm.
anaerobic. It also can be used to make amino acids.
Yeah.
So pyruvate can be used to make alanine,
Hmm.
right? Ultimately, they're both 3 carbons. Alanine's R group is just the CH3, just a little
Hmm.
little tiny R
Little
group.
carbon. Hmm.
Yeah, exactly. But they have the correct number of carbons, 3 and 3.
Hmm.
Although keep in mind, every amino acid is going to also have an amino group, right? We talk about the core components of every amino acid. You have an amino group, a carboxylic acid, the R group, and a hydrogen. So
Hmm.
we actually form alanine through what we call a transamination reaction. Just a fun fact, it's very
Right.
similar. Pyruvate and alanine, they're very similar in structure, although any amino acid is going to need a nitrogen containing amino group, so we'd have to add that on. But it could be
Right.
used to make amino acids. And
Hmm.
alanine, there from then on, can be used to make others of our
Hmm.
20 biologically relevant. Um, and then again, we can talk about if we have pyruvate, you can also go back up to glucose through gluconeogenesis if you need to. So
Right.
lots of different options,
Yeah, which
as
again is
always.
right, of course, right? We talked about the first branching point. This is another one, important one, and we talked about the different like different environments that the first two pathways would go. Do we have oxygen? Do we not have oxygen? But like these other pathways are pathways that happen more if we don't need energy. Once again, the other two,
Mhm.
like aerobic, like continuing with aerobic respiration or anaerobic, that's still when we lack ATP. Like if I lack ATP, of course I need to produce more energy. I either need to keep on
Mhm.
doing glycolysis or I need to continue on with aerobic respiration. But again, we have to think, what if I have enough ATP in the cell? What if I don't need any more energy in the cell? Well,
Mhm.
therefore, maybe I want to do something else. So that'd be like more producing the alanine, an anabolic pathway. Well, that means my cell already has all the ATP. So this is again going back to let's start building things. If I have enough energy, let's
Yeah.
keep
Why
building,
not?
right?
Yeah.
And again, the gluconeogenesis pathway is a very interesting one because that's actually something that's stimulated not when we are in a fed state, but rather in a
Mm.
fasted state, which is kind of an interesting idea. Glycolysis is upregulated when you have insulin, which is the fed state. Gluconeogenesis, taking pyruvate and bringing it back to glucose, is really stimulated in the fasted state. But you have to I always kind of like, I remember I had a difficulty with this at first, where it's like, well, if we're in a fasted state, why don't I just want to produce more energy in the cell? Like, why don't I just continue on and produce more energy in the cell? Why am I remaking glucose? Which is, first and foremost, gluconeogenesis is a very energy intensive process. Like, you have to input energy. But the thing is, we have to recall who's doing the gluconeogenesis, right? It's the liver, really mostly the liver that's doing gluconeogenesis a lot of the times because it's producing that glucose to be sent out of the liver for the rest of the body, right? Because this is something that's very important where gluconeogenesis is taking pyruvate and going back up to glucose, but only the liver has that last enzyme that can bring it back to glucose. We talk about hexokinase traps the glucose in the cell. No other cells can go backwards except the liver, right? And a little bit of the kidney, but the MCAT doesn't really talk about the kidney too much. It's really the liver. Now, like that's really important because imagine if we are in a fasted state, like I got glucagon, I don't have blood sugar, right? None of my other cells can actually release sugar back into the blood except the liver. So that's why glucagon is actually going to upregulate this reverse process because this is really happening in the liver to send the glucose out into the body, right? So that cells that or organs that need the glucose that are very important, like the brain or muscles, will have that glucose so you can keep on thinking and moving if you're in a fasted state. So this is one thing that I was always, I'm gonna be honest, I was so confused by this when I first learned metabolism. I was like, why when I'm starving, I'm making glucose? Why don't I just make more energy, right? But it's kind of conceptualizing that it's really the liver that's really doing this process.
Yeah. And you're bringing up something that I think is really fun and important to hit on. And that is there's a difference between fed and fasting and your cell has enough food. Like
Yeah.
food for the body is not the same as food for the cell, right? When we talk about being fed, we're talking about having a high or fed be high blood glucose. We're saying our body has a lot of energy that is usable but not usable for the cell yet, right? Which is part of why we bring up these different layers, right? If you have insulin signaling, you're going to make ATP. Your cell might not have that much to eat, might not have that much usable energy. Insulin is going to help them bring in glucose. Just because I have glucose in the cell doesn't mean that I have energy to use. Glucose is way too big and energy dense for an individual cell to use for a regular chemical reaction. So we have to break that down into forms that are usable for the cell. In that situation, when we're in a fasting state, our body is fasting. Our body needs energy, right? If we took that pyruvate and we just made ATP, your individual cell that this was going on in might be fed. But what if there are more important tissues in the body as a whole that don't have enough pyruvate, right? We don't need as much energy to be used in our skin as the liver, right? Or in the brain or the muscles, right? Like you have to recognize that the body will want to shunt energy to particular areas. So I like that you bring up body system wide, we're fed or we're fasting versus energetic requirements of a cell.
Right.
Because they're different. They should be in sync and coordinated, but they're not the same thing.
They're not the same. And that's why there's different signals for both. Like insulin glucagon is very much the body signal, like is the body fed, is the body fasted, whereas things like ADP and ATP, right, are more
Exactly.
the cell signal. Is the cell fed? Does the cell have energy? And that's why it's cool to think of those, especially when we talked about glycolysis, like, oh, what does insulin glucagon do? But also how does ATP, how does the amount of energy in the cell impact which pathway goes? Which
Yes,
is
I
really
love
cool.
that.
And of course, right, which is, I think this is again really goes back to the intention of the body, right? The body
Mhm.
is so common because we have to think about it on the cellular level, but also on the organ, organismal level. Am I saying that word
Yeah, there
right?
you go.
Level.
Yeah.
And they have these kind of two interplay, which is really, really cool. So we've kind of talked about we talked about the two kind of catabolic pathways, right? Which is pyruvate to aerobic, anaerobic. We've talked about if the cell has enough energy, and that's kind of that alanine. If the body doesn't have enough energy, we can go back and gluconeogenesis. And I think those are kind of the four main pathways pyruvate's going to go to. I don't think we missed any there, right?
No, no,
No,
no.
right? And so I think that kind of takes us to the next really important pivot molecule. And I think of all of the pivot molecules, I actually think maybe this is my subjective, but the most important
I think
pivot
so
molecule.
too. I
Because
think so
it's
too.
such a connector. Like, especially when I know we give this lecture all the time. Like, acetyl-CoA is what really connects so many
Everything.
like really
Pretty
everything.
much. Yeah.
Because acetyl-CoA is that really important molecule we talked about before when we talked about the PDC and TCA, that it's that product of the pyruvate dehydrogenase complex from pyruvate to acetyl-CoA. It's important that once we have acetyl-CoA, PDC, irreversible. We
Yeah.
are not going backwards. Now we have the acetyl-CoA, but that acetyl-CoA very flexible on what we can do with
Mhm.
it, depending, once again, cellular and body signals. What does the body versus the cell need? We talked about one of the pathways, which is the aerobic respiration pathway, which is going on to the citric acid cycle, producing energy, very catabolic, okay, the cell needs energy, but there are other pathways that acetyl-CoA can go into.
Yes. Yeah,
Right.
I agree. By the way, acetyl-CoA I do think is the most important.
Okay, we're on the same page on that
It
one.
is.
It's good.
Yeah.
Right.
I mean, and even like when I was talking about pyruvate and the different fates it could have, I'm like, well, pyruvate, the fact that it can make acetyl-CoA means that it's got a lot of different options. But of course, we've got a whole segment for acetyl-CoA. So we got to give it its time to shine,
Right.
right? Keep in mind that this is produced directly by the pyruvate dehydrogenase complex, but there are
Mm-hmm.
actually other ways to make acetyl-CoA, right?
Mm-hmm.
When we talk about using fats for energy, right? Specifically, let's say a triglyceride. A triglyceride is made of a glycerol backbone.
Mm-hmm.
So it's three carbons, three oxygens. It's like a linking molecule that keeps those three fatty
Mm-hmm.
acids together. Each of those fatty acids can produce a ton of acetyl-CoA through a process
Mm-hmm.
called beta oxidation, right? And we'll talk more about beta oxidation, but basically you could take this big long chain of carbons. It's a hydrocarbon with a carboxylic acid on one side, and you can just basically cut two carbons off at a time and make an acetyl-CoA. So we produce it through pyruvate or through the pyruvate dehydrogenase complex, but beta oxidation of fatty acids, so fat breakdown,
Mm-hmm.
also gives us acetyl-CoA. So this is our entry point, right? We've been talking this whole time about glucose metabolism,
Mm-hmm.
but fats also enter this pathway through acetyl-CoA. So we
Mm-hmm.
have more produced through beta oxidation of fatty acids. And then we also can talk about different amino acids that
Mm-hmm.
can be used to make acetyl-CoA. And these are called ketogenic amino
Mm-hmm.
acids. And there really are only two that can only do
Right.
ketogenic or that are exclusively ketogenic amino acids. And it's the L's,
Mm-hmm.
leucine and lysine.
Mm-hmm.
So unlikely that they'd ask you a question about that, but those are the only two amino acids that are exclusively
Mm-hmm.
ketogenic. And L for lipids is what I always thought.
Oh, cool. I like that.
Yeah. So
Mm-hmm.
we've got different things that can feed into acetyl-CoA. And then even from there, we have different options of what we want to do with it. We could
Right.
go straight through, like you said, TCA cycle. We could also, if we wanted to, if we
Mm-hmm.
had a lot of energy, we could take all these acetyl-CoAs and make new fatty acids.
Mm-hmm.
It depends. Beta oxidation, fatty acid synthesis. Are we fed? And therefore we want to store energy as fat?
Mm-hmm.
Or are we fasting? In which case, let's go ahead and use that
Mm-hmm.
fat or that fatty acid to make acetyl-CoA. And then there's an interesting thing about ketone bodies,
Oh, I love talking
which
about
is fun. And I
ketone.
know you love this.
Because like again, I mean, anyone listening for a little
It's
bit
cool.
knows that
Yeah.
we're really into both of us are really kind of into the gym space and then like the health space. So keto diets are always like that big thing that happened a while. I think it's still a pretty big thing. But
Oh,
it's
it's huge.
it's
People talk about it all the time.
still huge. Where when the body doesn't have sugar, say, because again, that's the whole idea of like keto diets, like don't eat carbohydrates, they're the enemy, right?
I
Only eat
know.
fats, right? There are
I love
some,
carbs.
yeah, me too. I know.
Love them.
There are some organs in the body that can't just like not have energy, right? Like take like the
Yeah.
brain, for example.
Mhm.
The brain's primary energy source is glucose, right?
She's very
So
picky.
very, very picky on that one.
Yeah.
But imagine if you're just not eating any carbohydrates. Like it's not like the brain's just gonna starve. Like we don't just
Oh, it's
die.
fine.
Well, yeah, that's fine. Yeah, I don't need sugar, whatever. It still needs to get fed, but if there's no sugar, there needs to be some other energy source. And this is where ketone bodies comes in and how acetyl-CoA relates. Because I want you to think of acetyl-CoA, we've just talked about before, super important energy molecule, TCA cycle. It goes, produces a whole bunch of energy, but a lot of the times we need to get this high energy molecule to organs that really need it, right? Like the
Mhm.
brain, like the muscles. So
Yeah.
I always kind of like think of ketone bodies as like storage like of acetyl-CoA, where we can't just ship acetyl-CoA into the blood. We can't just do that. What we actually do is we combine two acetyl-CoAs into ketone bodies. There are kind of three ketone bodies, only two are really usable. And then we ship those ketone bodies into the blood. It circulates in the blood. Those ketone bodies are taken up by those brain and the muscles and then converted into, guess what? Acetyl-CoA to be used for energy at those sites. Because again, if they don't have glucose, they still need energy. So that's kind of the key where acetyl-CoA, well, this is really, really sick, where we can use it for TCA cycle in the local cell, in the cell itself. We can use it well for in a fed state. We can use it for storage into a fat. But if the body is kind of starving, right? If the body is in a fasted state, right? It can say, hey, we don't have any blood glucose. We've done our best to try to replenish as much blood glucose. Instead of getting those glucose into the blood, now we need to go to the ketone bodies. And that's where acetyl-CoA can go instead. Because there's kind of these three pathways. TCA, fats, or ketone bodies. And I think the ketone one is really, really cool because it's kind of like, just think of ketone bodies as like stored acetyl-CoA that can go into the blood and to the target. Sorry,
Yeah.
I just went super, like, I was like, oh my gosh, I love talking about ketone bodies. So yeah, that's
You're
the
like,
kind
I
of like,
blacked out for a second.
sorry, I'm back now.
No, I love it.
And I think it's also really cool, and also to conceptualize the connection between like sugar, like carbohydrates and fat, is in this acetyl-CoA place. Because a lot of times I ask students like in class, like, imagine if you eat a bunch of sugar and you just are sedentary the whole day. What will end up happening to your weight? Obviously, it's going to go up.
You're gonna go
You're
like,
gonna start gaining weight, particularly in fat. But wait a minute, you're just eating pure sugar. How is it that you're gaining fat, right? When you're eating sugar, well, you should be eating sugar. Why don't you just gain glycogen and that's it? Well, it's acetyl-CoA. That's the key. Because at first, your body was like, oh, look at all this sugar. We're gonna keep on doing it, converting it to acetyl-CoA. Then it was like, oh, wait a minute.
yeah.
This is too much sugar, and we're not using any of it. Let's store it all. Take that acetyl-CoA. Your body's probably got so much insulin at that point, and saying, let's store this all and turn that acetyl-CoA into your fatty acids and store that as fat. And so again, I like using these kind of like, oh, we all know this in these pathways to kind of help make those connections. Man,
Love
I
it.
am just like ranting about this because I just love
But it's
acetyl-CoA.
good. It's
Yeah,
good.
yeah.
I love whenever I feel like it's very tangible when we get something that we're very excited to talk about.
Right.
And it always makes me happy to see you light up to talk about something.
So sweet.
Um, it's probably the nicest thing I've ever said to you.
I know.
Um,
You should see it before before the camera begins. It's the word.
Mike.
I'm just kidding. I'm just
Um,
kidding.
so as we beautifully just said, or as Mike beautifully just shared, right? We have lots of different contexts for how the body's going to use acetyl-CoA, right?
Mhm.
Am I going to be using this in an anabolic sense? Am I going to use this in a catabolic sense? If I'm stuck in a tough situation where I'm really out of energy, there's also that temporary sort of transit form of acetyl-CoA that we can use in other tissues. When we talk about fats specifically, because you're
Mhm.
bringing up this connection, which I love, if you're
Mhm.
sitting around just eating candy all day and you're not
Mhm.
moving, yeah, you're going to store that as fat. Again,
Mhm.
first instinct, you've got all this sugar. First
Mhm.
instinct is, okay, can we store it? Can we use
Mhm.
it for stuff? Let's
Mhm.
say we're already good. Our
Mhm.
ATP is really high. We've already done the cellular maintenance we need. Like, we don't need this right now. We've already stored everything that we can as glycogen. I like that you brought up that it's finite. We could make this into fatty acids and
Mhm.
then into fats. So again, just to recap, I know I mentioned it earlier, but a triglyceride that would be stored in a fat cell or in a adipocyte in our body is a glycerol backbone and 3 fatty acids. We could take acetyl-CoA and fuse those into the fatty acids themselves, right? Or we could take a fatty acid that exists and cut off acetyl-CoAs. Every process or every round of beta oxidation, which between the two, fatty acid synthesis or beta oxidation is probably tested a little bit more often. It's the breaking down of a fatty acid into acetyl-CoA. Every round is going to snip off 2 carbons of that. Let's say it's a 16 carbon fatty acid, which is the most common. If you cut off 2 carbons at a time, every time you chop off those 2 to make acetyl-CoA, you're also going to make an FADH2 and an NADH. And we say that happens in 1 round. Okay. The one thing that's important to know, and I've seen this tested so many times, is that if you have a 16 carbon fatty acid, you have the first round, you chop off an acetyl-CoA, you have now 1 acetyl-CoA, an NADH, an FADH2, and a 14 carbon fatty acid. Chop off another molecule, right? Now you have 2 acetyl-CoA, 2 NADH, 2 FADH2, and now a 12 carbon fatty acid. That's going to keep going until you have a 4 carbon fatty acid left. And the thing that's important to know here is when you have that round of beta oxidation that would chop off another acetyl-CoA, you actually end up with 2 acetyl-CoAs at the end. So a 16 carbon molecule will make, or a 16 carbon fatty acid will make 8 acetyl-CoAs. Each of them has 2 carbons, right? 16 divided by 2 is 8. But there's actually only 7 rounds that would be used to go from a 16 carbon fatty acid to 8 acetyl-CoAs. Because that last time you cut, you cut it in half and make 2. So just so you know.
Super common mistake. I'm so happy you brought it because I've seen that mistake so often, right? You would think, oh, it's 16 carbon, right? Oh, that means 8 cuts. No. Right. And I actually recommend everybody, guys, draw a 16 carbon molecule and cut. See how many lines you can cut. And to make these 2 carbon molecules, what you'll find, you'll only cut 7 times because the last cut produces 2. It's not like you have to make another cut to produce the last one. So, put it wonderfully where 16 carbons, 8 acetyl-CoA, because again, the carbons aren't going anywhere. It's still 8 acetyl-CoA, but only 7 cuts, therefore only 7 NADH and only 7 FADH2 produced as a result of those cuts. What's also cool though, it's not only the fatty acid that can be used for energy, like when it comes to fats, but also the glycerol. I know you like talking about this one, so I'll leave you to talk about it. The glycerol can also be, we're using all the energy from our fats, so not just from the fatty acids. So how are we going to use that glycerol for energy?
Think about what it sounds like, right? Glycerol. I said earlier, we have a triglyceride. This is the backbone for a triglyceride. So there's 3 fatty acids. They need to be linked. This is the 3 carbon molecule.
That's what
sounds like glyceraldehyde. Thank you. Took some wheel turning. Yeah, glyceraldehyde. They sound very similar. They can be, glycerol can be converted into glyceraldehyde and then isomerized into DHAP and used for whatever you want it to do, right? It could be through glycolysis, and we could go proceed and make this into ATP through aerobic respiration. We could also go gluconeogenesis with this as well. Because again, we are creating a glycolytic intermediate. We have both options depending on the cell type and depending on the need. So yes, glycerol can also be used as an energy source. Again, your fat, your triglycerides really are pure energy, basically.
That's much energy.
That's why,
Yeah, and I think it's also cool because they're actually I think they're actually even more energy dense than glucose. Like fats are actually just even more, we get more
gram,
energy per gram, but our body still, like, its primary energy source is, if it's glucose is available, it's going to use it first, even though technically fatty acids do have way more energy in them. So maybe that's the argument from the keto people to be getting their energy from fats, but we'll leave that for Reddit or something. But that's cool. So again, coming back, so acetyl-CoA, we just talked about how flexible it is, how it connects everything, how you can get acetyl-CoA from a number of sources, from aerobic respiration, PDC, from fats, and also from ketogenic amino acids, and how it can go into and become fat once again. It can continue in the TCA, or we can use it for ketone bodies. And so this is kind of like, again, the flexibility. But one thing to keep in mind that we've mentioned before, what pyruvate cannot do What it
Yeah.
cannot do.
Or what acetyl-CoA can't do.
Sorry, I might have said pyruvate. Acetyl-CoA. What acetyl-CoA cannot do is go back to pyruvate.
Yes.
PDC is irreversible. I think that idea is tested so often on the MCAT. Like people
Yeah.
think, oh, I have acetyl-CoA, it's just going to go back to pyruvate. Does not work like that. Once you're
Yeah.
acetyl-CoA, it's stuck that way in the pathway. It can do a whole bunch of stuff, but what it cannot do is go back to pyruvate is the key.
Yeah. Yeah. And again, it kind of makes sense. Like our body stores fat, and we talked about why it is more energy dense. But also, like, if it was hypothetically possible, why wouldn't we just have a ton of glycogen? If that's our body's primary interest, like, why not just not have any fat and just have a ton of glycogen? That'd be even easier, right? No, that's not how that works.
That's not how this works. All right, glycogen is actually like really not space conservative. Like it's actually
widespread.
It's very widespread, whereas fats stack so well on top of each other, so it's actually a way better storage. And the body is so wise, the body is so smart. Like when it comes to that sort of stuff, I know evolutionary I knew you were gonna say. I was like, evolutionary biology has kind of come up, where it's kind of like, yeah, maybe early organisms started with like glycogen, but then once we found that, like, again, evolutionarily, like the fats were just a better storage method, that became our primary storage method of energy. And that's why when we have excess, it's fat that we build up a lot of, not glycogen. Right? I think it's cool. And I think that kind of, now that we kind of really hit on acetyl-CoA, there's kind of like one more kind of intermediate that we've already discussed before. That's been
all of these we have,
all of these we've talked about before. We're not really in this context, which is oxaloacetate, right? Which is also a lot. Again, and even coming from me personally, I think the first three are ones that I always think of when it comes to pivot molecules. But oxaloacetate is also a really cool molecule because it's also used in a few pathways.
Yes. Yeah, oxaloacetate, I feel like gets no love. Oxaloacetate, if you have not seen our other episodes, it is the starting/end product of the TCA cycle, right? It's that 4-carbon intermediate that's going to bind with acetyl-CoA to make citrate, which is the citric acid cycle. And we know it from the TCA cycle. So we know that that's part of what it does, is it can be used to take acetyl-CoA and help break it down to ATP, NADH, FADH2, all that stuff that could be used for energy for the cell. So that's certainly one thing that we could do. And that's what we're used to. That being said, we also see oxaloacetate in gluconeogenesis, right? Keep in mind, gluconeogenesis, glucose, new, genesis, make new glucose. We're not going exactly the reverse of glycolysis, right? There are other enzymes that we need, right? Oxaloacetate is an intermediate of one of those other enzymes. So that is also something that we see there. And it can be involved in gluconeogenesis. Typically not something we want to do because we want to keep our TCA cycle functioning very well. But if we truly needed it, you could pull oxaloacetate out and use it for gluconeogenesis. Very much a last resort, but that's part of it. So it's a intermediate in gluconeogenesis. If we truly need glucose, we can pull that out a little bit. And it also can be used to make amino acids as well. There are some amino acids that can't be made from alanine, right? And so specifically, oxaloacetate can be used to make aspartate, and reverse. Aspartate can be used to make oxaloacetate. So it's another opportunity for amino acids to be involved in metabolism, either on the forming side or on the breakdown side.
Right.
Which is pretty fun.
Which is pretty fun, right? Because again, I know when I first learned I always just thought oxaloacetate, that's part of the TCA. It doesn't really do much else. But yeah, realizing it's an important gluconeogenesis intermediate. Also, it's another kind of entry point for amino acids in this cycle. So yeah, I think oxaloacetate is a great connection as well. Again, in terms of like testing, I don't think like oxaloacetate gets tested as much as like acetyl-CoA or pyruvate, but definitely testable material. They're 100%. And I think in terms of the kind of intermediates, I think those are kind of like the four we covered. First one, which is G6P, that really important, can decide a whole bunch of pathways. We talked about pyruvate, right? Making like, again, is a really important pivot molecule. We've talked about acetyl-CoA, an important pivot molecule, depending on the environmental conditions. And finally, oxaloacetate, where it has a number of options as well. And again, the goal that by the end of today, that I hope everyone listening has realized, is that it's not just pathway X, and then here's pathway Y that's completely separate. No, they all interweave together with aerobic respiration as the central kind of like pivot. Like aerobic respiration is the center, and then everything sort of branches off at these kind of really key pivot points. So understanding the importance of these pivot molecules, that they can go into a number of directions depending on what the cell needs, I think is critical when
Yeah.
studying metabolism. Something I'm so happy that we had a chance to talk about.
Yeah, it's almost like the tree of life, right? You have aerobic metabolism as the trunk, and you got all the branch points coming off. So it's very easy to feel very overwhelmed by metabolism because there are so many different processes, but I want you to visualize it in this way. And of course, our aerobic metabolism steps are really the ones that are going to be tested the most frequently, right? So glycolysis, the pyruvate oxidation with the pyruvate dehydrogenase complex, the TCA cycle, and then oxidative phosphorylation and the electron transport chain. Those make up the core catabolic pathways, right? We also talk about the other accessory pathways, and I'm just going to mention them all the others by name, so that you know if you're going to go study metabolism, these are all of them that you're responsible for, at least the ones that show up frequently, because I always personally felt even though I knew glycolysis, I was going to forget something, right? Glycolysis, gluconeogenesis, glycogenolysis, and glycogenesis, right? We've got pentose phosphate pathway. Those all are kind of relating to glycolysis and glycolytic intermediates, right? We then have pyruvate oxidation. We have beta oxidation and fatty acid synthesis. We've got the TCA cycle and ketogenesis. We've got fermentation. Should have mentioned that earlier. The electron transport chain and oxidative phosphorylation. Those are all the processes that you are responsible for on the MCAT. Mike, did I leave anything out?
I think you got them. I think I was like following along. I'm pretty sure everything I would say. Do we say fatty acid synthesis like with
Yes.
Okay, then yeah, I think that's everything. And again, but what's cool is like everything that you just mentioned fits into our pathway. Like we've talked about every single one. We've already mentioned them all. So and I actually really recommend this, and I think you have a really great one that's actually as part of our course, you can even see it, where make a map of all your metabolism. Like actually, like if you're listening to this and you're struggling with metabolism and connecting this all in your head, like we've maybe been listening and you're like, oh my goodness, it's hard for me to visualize. Draw a picture, make a metabolism map with aerobic respiration as your central pathway, and then actually draw it out. Where do those pivot points happen? What do they become? And then honestly, I would also recommend checking it with like an actual, like there's so many of those resources out there. Like once you make it, like do it your best to do it on your own and then check it and see, okay, where it is. But by doing this, you're actually physically making these connections. So it helps with encoding, so that you can actually really understand them, and then you can apply it once you understand it.
Yes. And as someone who's made one of these maps before, and by the way, if you're a course student, I have a whole lesson, a mini lecture on metabolic connections like this. Start with the pathways. Again, you could use the tree trunk analogy, get all of the key intermediates that we just mentioned on there, right? And then start drawing the branch out points. You do not need to put every intermediate and every enzyme of, let's say, glycolysis. It's going to get way too complicated because every one of these processes has different steps and enzymes and regulation. No, keep it simple. But if you know these pathways individually in terms of like their actual flow, where in the body they happen, where in the cell they happen, what, key points of regulation are, what you need to do is put them together. And that's a great way to do it. I love that as kind of call it a homework assignment. Call it a suggestion. I don't think we can give you homework, you guys.
You can do whatever you want, but we think it'd be a great idea.
We can strongly suggest something that would help you a lot. And again, also practice. Lots and lots of practice, because again, most of the time, these pathways are not just tested like, what is the 8th step of glycolysis? It's if I inhibited this thing or under fasting conditions, what would happen to the activity of this? It's all very interconnected. It's very based on the connections, right? Like we just talked about. So super excited. I'm glad that we were able to kind of close up our metabolism unit. And we are ready to move on. If you have questions for us about metabolism, as always, come see us in our live sessions, right? Or leave us a comment if you have anything that you are curious about that we did not cover, because I know that we love to help you guys with whatever kind of content or strategy or MCAT relevant, whatever. Tell us if there's something you want to see on this podcast, because we're always looking to help you guys, whatever that may look like.
Absolutely, absolutely. Yeah, I know we've got with this closing off the unit, I think we have like one more like kind of like topic that we really want to hit on. But then after, like I think we need to meet to kind of like figure out what we're going to be doing. So this is if you're ever like I really wish that Molly and Mike really just hit on this topic. This is the episode to do it. Comment like what you want to see. Come see us in our free trial sessions, or if you're a course student, even like come to our office hours. Molly holds a bunch, I hold the course, right? And let us know. We're kind of receptive to it. And then we'll plan it from there. But I think the next episode we have something kind of very particular planned because I think it's very cool to know these pathways. But I like kind of thinking about like how did we figure this out? Like how did we actually like
study this stuff?
how did we actually study it? And it's funny because I actually like myself like studied a lot of like maybe not in aerobic metabolism, actually like more cell cycle pathways. But there's all these methods that we use to study proteins, to study intermediates, and that's what we're going to be talking about next episode. We're going to be really diving into like analytical techniques. We're going to be talking about how we actually study these things, interactions between molecules. How did we figure out these things bind? And see all the analytical techniques that the MCAT will expect you to know. So I'm kind of it's like because I spent years doing this stuff, and so it really like it was so painful in the time because in theory they're cool, but in practice they're the worst.
In a
Don't get
lab,
me
not as fun.
in the Western blots, don't get me started, but we'll talk about those next episode. But I'm really excited to talk about the next episode.
Me too.
Yeah.
I'm excited to see you get really excited about it.
Excited and like a little bit of like
The dreading,
shock.
yeah,
Dreading.
flashbacks. Oh my gosh. Well, tune in next time, you guys. It was so lovely to have you. I hope you enjoyed our metabolism unit. This was a really fun one to make for you. Let us know if you want anything covered specific. We love to hear from you. And until then, happy studying. Have a wonderful rest of your day, and we'll see you soon.
I know.
Bye.
Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
And Molly Hudash.